Thank you. Hello, everyone. And today my topic will be the geometry of creativity, or understanding generative models in brains and machines. So at Kempner Institute, our goal is to understand the natural and artificial intelligence and let them to inspire each other. So on a fundamental level, what type of intelligence do we really care about? We know that computers can already solve equations, memorize text, retrieve information much faster and reliable than human beings. But still, we don't think they are intelligent. One reason is, in our wildest dream of artificial intelligence, we want to display the level of creativity as any genius among humans. We care about imagination, the creation of arts, creation of mathematical theories, or creation of poems. And this endless creativity defines our humanity. So the ability to come up with solutions that nobody has seen before is something we really care about. What's the path towards this type of future? So in recent years, we have seen promising results in the field of generative modeling. So generative models is a type of machine learning models that learn from the distributions like photos or text, and then create new samples from it. So one classic example is GAN, or generative adversarial networks. So these are models that are trained on image nets and to create object images from it. And here are examples of GANs trained on human faces. And you can say they create photorealistic face of human that doesn't exist. In the last 2 years, diffusion models have been catching up and becomes highly popular in last year due to stable diffusion and DALL-E tubes. So we have seen them to synthesize amazing combinations of styles and contents using just text prompt like this. Going further, in last year, we have also seen ChatGPT, which can write you a poem about intelligence in the style of Alexander Pope, the Harry couplets. Or if you like, you can also write you a poem in the styles of Donald Trump's speech. So these are all very interesting. But are these models paving the path toward human intelligence or something aliens? We don't quite know the answer until we understand these models deeply. So in this talk, I will show you how we can start a journey of understanding generative models deeper. And when we have a deeper understanding, this could help us to also understand the representations in the brains. And this exemplifies the virtuous cycles between AI and neuroscience. So in the first part, I will specifically tell you how the diffusion models decide what to create. And this is a joint work with my friends and colleagues, John Wastala from Harvard Medical School, and it's called the geometry of diffusion models. So why do we care about diffusion models specifically? Because it is leading the charge in generative modeling in many fronts. We have seen state-of-the-art results with diffusion models for image modeling, 3D shape modeling, or even video modeling recently. And they have won prize in art competitions that made the art community unhappy. So maybe not everyone is familiar with the math behind diffusion models. So here's a quick introduction to it. On the basic level, we are interested in modeling certain distribution, like the distributions of dogs. And this distribution, we only have samples, but we don't know how to sample new examples from it. But there are some other distributions like Gaussian noise, which we know how to sample arbitrary sample from it, but it's not so interesting. The idea of diffusion model is to find links between these two distributions, the clean examples and the pure noise. There's an easy way to turn any example into noise just by adding noise to it and decreasing the signal. We call this the forward or noise process. If we know how to reverse this noising process or how to denoise these noisy images, then we can just sample a noise pattern and turn it into data. In diffusion models, this denoising part is done by neural networks. So they are trained to predict the noise part from a degraded image. Given a noise sample, the neural network will predict noise patterns, and by iteratively subtracting this noise pattern, you can denoise the noise sample and arrive at a clean sample. And mathematically, this neural network is approximating the time-dependent score function of the data distributions, and we'll use that in the future. So first, let me take a look at the generating process in action. As we can see in the math, the diffusion process is a time reversal of a noising process. So it's not so surprising to see in the generating process, the latent states stay as noise, and gradually towards the end, some clean images is revealed. Similarly, if we see a model trained on faces, you see a similar trend. Even for the most recent model, like Stable Diffusion, you see a similar phenomenon, which is the well-formed images are unveiled towards the end. So although these models work amazingly, their painting process seems pretty different from the artist. The artists are definitely not just subtracting noise from a noise pattern. They do so by sketching the overall gist, shapes, and then followed by details. So you already have a rough ideas of what they are painting at first and then refine them gradually. So we are curious, how different are they? Are diffusion models really entirely different from the creation process of artists? So to understand this, we first take a deeper analysis of the generating process. We examined the next layer of paint that puts on the canvas. What we mean by that is we take the difference of neighboring state during diffusion, and then we normalizing them properly. So even if this layer of paint is faint, it represents something that is highly interpretable, which is the average phase for a phase generating network. Moving forward, we see very similar phenomena where the difference between the neighboring states are an average phase, and this phase image becomes clearer and clearer. If we zoom out a little bit, during diffusion, this normalized state difference will first look like an average phase or rough sketch of the example. And then you see it's getting refined and approaching the final sample as you see in the last step. But towards the end, the sample is contaminated by more and more noise until you cannot see the sample anymore.
So Binxu,
yeah.
Can I just ask a question? So
yeah.
What is the time schedule here is uniform? So how, I mean, in diffusion, you can schedule the time and the amount of noise in very different ways. So
yes. Yeah, so currently in all these plots, we just follows the normal schedules that are used in the pre-trained models. And so if we do the math, like the noise or the beta follows like a linear schedule, and alpha is like exponential decay, I think. Yeah, we basically followed the schedules used in the models themselves. And in this plot, like all these grids are like equally spaced in time steps.
And, you say in the right thing, you call it contaminated, but is it really maybe just the point that basically at that point you basically got the right image almost correctly, and all you need to do is fix a little bit of the remaining noise, so it's not maybe contaminated is the right word to say?
Yes, you're exactly correct, and we will show that in the next slides. Yeah. So this illuminates that the generating process is not as we see in the naive visualization of the states, but it has a more gradual generating process which we can interpret. But towards the end, this is not so interpretable. So we search for a better way to visualize this diffusion process. And digging into literature, we find this statistic called an estimated outcome. So it is a linear combinations of the current latent states and the score function at this state, and normalizing them properly. So you can think about it like a linear teleportation from your current state directly towards the end. If the data set is just a delta function, this will always point to the correct data. But if it's not, something interesting may happen. So we visualize the statistics throughout the diffusion process, and we see an even clearer picture of this generating process, where at first it's very similar to a state difference or the moving direction of the states. And towards the end, it resembles the actual states we are seeing. You can see it start from average sample and then specifying like gender, hairstyle, face orientation, these high-level variables. And towards the end, it adds a fine-grained, high-frequency skin texture details. So in some sense, if we look deeper, the diffusion models do create a rough sketch and then refine it somehow like an artist. We think it's not enough to just visualize the states. We want to understand the geometry of it. So we take the trajectories of the states and find it's almost like two-dimensional curve in the high-dimensional space, like 3,000 to 300,000 dimensions. The two top PCs explains 99.9 variance, and the space spanned by the initial noise and the final state explain more than 99.5% variance. Moreover, on the plane spanned by the initial states and the final image, the dynamics is nearly a rotation, and it's well fitted by rotation equation like the following, which explains more than 98% of variance. So observing all these curious phenomena, we desire a theory to explain how the initial noise pattern is related to the final samples, and also to describe the dynamics of the latent state or the estimated outcome. And finally, to predict why some features are specified earlier in the estimated outcome. To do this, we first build a simplified model of the image manifold, which is just a degenerate Gaussian distribution, which has a mean and a covariance. We expect the mean to be like a mean image, so we can expect like an average face for face dataset. And the covariance can be factorized into the eigenvector and eigenvalues. And we think the eigenvector spans the so-called signal manifold or data manifolds. And the orthogonal space of it is like the off manifold or noise subspace. On the signal manifold, there are features with smaller variance or smaller eigenvalues, and features with larger variance or larger eigenvalues. So for images, from the study of the image statistics, we know that low frequency layout information usually have larger variance, and high frequency details usually have smaller variance. So we can keep that in mind. Given the setup, we did some analysis of the diffusion sampling ODE, or the probability flow ordinary differential equation, which is writing at the following. And we take all the terms into the place, we get this. And we find we can redefine the variables by shifting the states and measuring distance to a scaled mean. Using that, we will arrive at a linear dynamic system. Further, we also find that we can simplify this using the Woodbury matrix inversion trick, which gives you the linear time-dependent linear matrix always have the same eigenbasis, which means that the dynamics along each of the eigenmodes are independent to each other. To not bother you with more technical details, here's our solutions to this equation. Basically, the solution has three terms. The first term is the scaling of the means or gradual adding to the mean. And second term is the off-manifold component. The dynamics is specified by this psi function. The third part is the dynamics of each of the feature components, and each of its dynamics is specified by the psi with different arguments of the variance. And we plot this special function here. So it has the following dynamics. The off-manifold components, which is this brown curve, will decay to 0 towards the end. In contrast, all the unmanifold featured components will start from 1 and then gradually converge to the standard deviation of that features. Depending on the variance, if it's smaller than 1, it will decrease gradually. If it's bigger than 1, it will increase. So we see that even the high variance features are added to the data very early in the process. It's kind of hidden by the noise, which is not reduced until the end. So this kind of explains, we see in the large period, we only see static noise in the diffusion state, but towards the end, the signal is unveiled from it. Using this solution, we can also explain why we see a two-dimensional rotation dynamics. So if we take this limit, which is a very good approximation, we will see that the state can be expressed in the following way. And this term, as we showed, it's very close to 0 throughout the diffusion process unless some time steps. So if we take this limit, then the hidden states can be expressed as this rotation dynamics equation, which explains the observations we see. Also, we analyzed the expected outcome or x0 hat. So its dynamics can be expressed in the following interpretable way. It has a term which is the mean of the distributions, and the dynamics of each of the feature dimensions is specified by this chi function. Its dynamics is plotted here. So all of them start very small. So it explains the estimated outcome. So they always stay on the data manifold and not contaminated by the noise of the off-manifold components. And because they all start very small, explains the estimated outcome usually start from the center of the distributions. And then each of the components gradually converge to a corresponding standard deviation in a sigmoid fashion. The large variance features, like the purple curve, will converge earlier, and the small variance dimension starts to change very late and converge later, which is the blue curve. If we take the derivative, we will see that this each of the dimensions change the most at different periods. The large variance dimensions, like the purple curve, are changed the earliest, and the small variance change the latest. So this explains our visualization of the projected outcome, where it starts from the mean of the distribution and gradually specified features from higher variance to lower variance. And we can say the feature development is ordered by their variance. So the take-home picture for the diffusion model thing is the following. So if we imagine the data lies on certain data manifolds, your initial point, your initial state is usually a noise pattern which is out of manifold. At this point, you can see no images from it. But it has a projected outcome which lies on the data manifold and looks like the average of the image. And during diffusion, this estimated outcomes first moving towards the high variance dimensions, specifying high-level features like layouts. And then it specifies the smaller variance features like the textures and arrive at final results. We think it's not enough to just have qualitative agreements of our series and the trained diffusion models. We want to see how well they predict the behavior of actual diffusion models. So for any image data sets, we can take a trained diffusion models and also take the image data sets and compute the mean and the covariance by principal component analysis. And then we can draw analytical solutions of the XT. So start from any initial noise sample, we can first draw solutions using the trained score networks, which arrive at a good image. And then we can use our solution to predict another. So where does it arrive at? For this model trained on CFAR images, you can see the actual trajectory converge toward a horse. And Surprisingly, our Gaussian theory also predicts something similar to a horse. And in the initial time steps, the agreement is good. And in the second half of it, the layout is still similar to a horse, but the high-level details, high-frequency details are not the same. We quantify the deviations of trajectories and find that after like 20 or 25 time steps, the trajectories start to diverge and have more and more error. We see similar stories for the face datasets. This is generated by diffusion models, and the Gaussian series can also predict like the first half of it, and you see a roughly good agreement on the high-level detail, high-level like layout information, like the hair here. And it also diverges after like 20 time steps. So we think the low-frequency layout information can be well predicted from the initial noise by our theory, and early trajectory is well predicted by the theory. So you may ask, why does it fail towards the end? I think the direct answer is it violates a single Gaussian assumption, because image is not Gaussianly distributed, so there may be multi-modes. And also, maybe their images are not a Gaussian on a linear space, there are nonlinear manifolds in it. So when it travels, you should change your mean and covariance as it travels. So we also have some data on the perturbations, but I don't have time to get into it. And here's the conclusion of the first part. We find that the trained diffusion models have gradual generation behaviors from layouts to details. And this links to the generation to some models like progressive growing gains. We see that the initial phase of diffusion is well approximated by a Gaussian analytical series without any neural networks. And this could shine light on how to make diffusion train faster or move faster. And low frequency layouts can predict it from noise. And finally, this provides some clues to the mystery of low dimensional image manifold structure in diffusion models. And this concludes the first part. If there's any questions I can answer, I can.
Any questions? Maybe one. So, yeah, go ahead.
Yeah, I mean, so you made the point here that it's sort of building on an initial scaffold and then adding details and so forth. But I thought when you actually analyzed the movement, you saw that that the noise collapsed late, but in fact the fine features were actually there early on and hidden by noise. So which one is actually the conclusion in the end?
That's a very good question, and so we personally think this picture is more correct to us, where like the high level or low frequency informations are added early on, and the high frequency or low level details are added to the end. But because of the dynamics specified in the diffusion models, it's just not visualized in the latent state. But using other statistics, you see the gradual adding of informations. Yeah.
Okay, thanks.
Thank you.
I was wondering, is this something you could potentially test out? Let's suppose that say high frequency information is, I don't know, whether the guy has an earring. Is it possible to test out when is it possible to add an earring to the guy at which stage of training or of inference, sorry, of sampling?
Yeah, that's very good points. And we also have the ideas of seeing that what type of information are specifically added to the image throughout the diffusion process. We haven't have a like high throughput way to like quantify this, like find the earrings and find this, but currently we just do it by eye. But if we have like a more automatic method to quantify that, we can see the times of what type of information is added like in real diffusion process.
And yeah, and I guess you have seen these kind of things. I mean, faces are very, very low dimensional, in some sense very
Yes.
simplified, but you have seen these things also in other data sets.
Yes. So like for church,
is the better is a better explanation there than.
Yeah, it's a very good point. Like we've seen this phenomena in all the diffusion model we analyzed, like for MNIST or church or something like that. But as you pointed out, like phase manifold, the manifold of phases, we think it's like more connected. So the mean of all the phases is a well-formed phase, but the mean of all the churches may not be the well-formed church, or the mean of all the digits is not the digit itself. So in that terms, we don't see as clear signals in the initial steps for MNIST or church, but for phase, we see a very clear formed average phase.
Mm-hmm.
And even for like stable diffusions, we see something very similar. Like you start from like a rough sketch of the outcome and then gradually add details throughout the process.
Mm-hmm. Thank you.
Thank you. Yeah. Okay. For the second part, for the sake of time, I will dive into the second part. So in the last part, you have seen we can understand diffusion models deeper. And next, we will show generative models are not just interesting per se. These models can also help us to understand the brains. And I will also demonstrate the effects of geometric perspective. This is a joint works with my PhD advisor, Carlos Ponce, and some of it is published in this recent paper. So why do we care about generative models in visual neuroscience? If we take a step back, in sensory neuroscience studies, we will take this paradigm, which is first you choose a set of images that neurons may be interested in and record a neural response to them. Then we infer the neural response based on the images and responses. So the problems for visual neuroscience is that the dimensionality of image space is so hard. So to study neural representation, the first challenge is to choose the stimuli and choose it wisely. The desired property of stimuli are they should be naturalistic, and they also should activate the neurons. And experimenters want to easy manipulation of them. In the history of neuroscience, people have developed many different image spaces for neurons in the ventral stream. For example, for V1, people developed Gabor filters or oriented bars. For V2, people developed textures. For V4, the contours or orientations. These are parametric low-dimensional image spaces. And for high-order areas, people also use objects or natural image samples, which are discrete and it's hard to sample like densely. So how should we go beyond the manual design of stimuli space? One idea is to let the neuron tell us what stimuli we should use instead of choosing that based on prior. So this will need two components. One component is the efficient sampling or optimization algorithms. So usually we use evolution algorithm for it. But these ideas of optimizing stimuli has been there for a while, but it didn't take off until recently because it doesn't have a compact parameterization of image space. This is where the generative image models comes in handy. So in the last 10 years, the GANs has been well trained and becomes a good model of natural image manifolds. These models will take in the latent codes or vector and then project them into an image space. This mapping is smooth, means that if you smoothly varying the latent codes, the image will also vary smoothly. So we think about it like a smooth parameterization of a vast image manifolds. There's still some challenges for this project, which is evolution search in image space, because the dimensionality of the search space is still very high, and there's stochasticity in neuronal responses. And in the end, as any experimenter to know, we have limited time with the neurons we have. So throughout the PhD, I developed a few techniques to do this better. We first do it by characterizing the geometry of the GAN image space and finding the important moving directions in the GAN image space. And then we developed the more efficient search algorithms guided by the image space geometry, which are published in these papers. So how does this work? So how to let neurons guide image search? And this is a work done by friends and colleagues, Wu Xiao and in Mark Livingston and Gabriel Kreiman's lab. In this experiment, we will first start from a bunch of latent codes, and then they're mapped to images by image generator. Then these images are showed on the screens, which are presented to the monkeys, and we recorded the neuronal responses in the monkey's brain using electrodes, and the fire rate of neurons are feedback to the optimizers, and the optimizer proposes a next bunch of latent codes, and this iteration will continue. So what does this tools brings us? The generative networks bring us a high-dimensional naturalistic image manifolds. And the evolution experiments empowered us to let a neuron guide the exploration of this manifold without the constraints of human prior. So now we are equipped with tools to study the neuron tuning on a high-dimensional image manifold. And it's useful to consider the neuronal tuning over the entire image space. We call it the tuning landscapes. On this picture, the evolution trajectories will be a path climbing the landscape towards a peak, a potential peak. So it's interesting to explore on the peaks and see the shape of tuning over there and validate it's a real peak. And also evolution trajectory will carry information about the global geometry of this landscape, which we can extract from it. So our research questions is, given this setup, did the neuron really find the peaks through evolution experiments? Sorry? I'll continue.
Okay, continue.
So, okay, cool. So we're asking, did the neuron really find the peaks through evolutions? And what do the tuning landscape of neurons look like around their peaks? And finally, what are some global characteristics of the tuning landscape we can learn from the trajectories? And how do they change over visual hierarchy? In this experiment, we will first start from the random direct position, which is like a random set of images, and then using evolution experiment to climb to a peak. And then we look around by manipulating that image in so-called manifold experiments. In this experiment, we were doing the following. So in the evolution experiment, we get latent space trajectories like this. And we will perform the principal component analysis of it, where the PC1 usually represents the main traveling directions. We will build a hemisphere around the PC1 using two other tangent vectors to it, usually PC2 and 3. And we will sample images on this hemisphere. We do hemisphere more because the generator or GANs has a spherical geometry. And we sample the azimuth or elevation grid on this hemisphere. And this is our geometric setup. We collect data from 2 monkeys and in 3 visual areas along the ventral stream, the V1, V2, V4, and inferior temporal cortexes. So what does the result look like? During the evolution experiment, you see, as we expected, the optimizers will update the images while the fire rate of the neuron will go up, as you see in the spike rate here or in the summary plot here. And finally, we arrive at certain images that resemble some parts of like a monkey or ear. After the evolution experiments, we conduct a manifold experiment. We retake this image and morph it along the 2-dimensional latent space grid. And we show all these images to the monkey and collect their responses. We see these smooth 2-dimensional tuning maps on this 2-dimensional hemisphere. We tried other hemispheres spanned by other PCs or random vectors, and we usually see very similar phenomena. Where we see a peak standing in the center of these maps and gradually decays when you move away from the peaks. And same story for random directions. Taking this together, it shows that we seem to really arrive at the local optimums in these landscapes. And tuning maps around these peaks are generally smooth and bell-shaped, which reminds of a radial basis function, or like a place field on an image manifold. Taking all the data together, you see they mostly share the same structure with a peak in the center and smoothly decay away. And we collect the data from 3 visual areas in the 3 panels, and you can see there's some qualitative difference among them. And that will be the topic of the next sections. So how does the tuning landscape change across the visual hierarchy? One simple thing we can quantify is the tuning width. By plotting, we will see that the V1, V2 arrays neurons will have a broader tuning peaks, while ITs has a much sharper tuning peaks. We quantify that by fitting a spherical analogs of a Gaussian function, which is a Kent function. And ITs will have the higher kappa value, which signifies the sharper tuning peaks than V1 and V2. We're curious if this phenomena exists in artificial visual hierarchies. So we did the same experiments in silico for convolution neural networks and see the same phenomena, where the initial layers has a broader tuning peaks around the peaks found by evolutions, and the last layer has a sharper tuning peaks. So you see an increase of the sharpness value along the hierarchy. As we mentioned, the evolution trajectory also carries some landscape informations. So we want to extract that by analyzing this trajectory.
Just a question, like if I'm thinking of like a mathematician, basically is what you're saying is that the Lifshitz constant is larger in higher layers than it is in smaller layers?
Yes. Because like one complication is that we only have 2-dimensional like submanifold around those points. So maybe there are like sharper directions in some uncharted directions, but in the data we get, it is so, yes.
Which is kind of very natural because it's more nonlinear, right? It's higher the degree, say, function, so it's likely that it will have a larger Lifshitz constant as you go as you go deeper.
Yeah. And it's tuned for like in visual neuroscience perspective, it's like more selective or sharply more specific to that specific combination of things.
Okay. Thank you.
Yeah. So the first analysis we've done for the search trajectory is to quantify the convergence time. So we see that for the primary visual cortex V1, the convergence time is much shorter than the time for IT to reach the peaks. We quantify that using the time to reach 63% of the total activation increase. And it finds an increase along the visual hierarchy. So IT cortexes definitely need longer time to optimize the features they want to optimize. We also find that for hierarchy in silico, which are convolution neural networks, from the first layer to last layer, you see the convergence times gradually increase. And you can argue that for FCA8 or the final layers in AlexNet, the evolution hasn't converged. And we quantify that and see the same trends as in vivo, the increase of the convergence timescale. Finally, we are interested in probing the required search dimensionalities. So we perform a so-called reduced dimension evolution experiment, where we perform 2 threads of evolution, one in the full 40,000 dimension space and one in a random 50-dimension subspace. We will see, can it reach any optimums in the random sections of the full space? And we curiously find that for V1 cortexes, the difference between the 2 threads is minuscule. And there you can find peaks even in a random 50-dimension subspace. But this difference increases throughout the hierarchy, and it finds in IT cortexes, the difference is largest. We quantify them by the activation ratio between the 2 threads, and you see that IT has the lowest ratio. Similar findings for the in silico models, that the larger and larger gaps are seen between the 50-dimension subspace and the 4,000 dimension full space. Putting this all together, we see three intriguing progressions along the visual hierarchy. We see the tuning width of the peaks decreases, and the time to reach a peak increases, and the required search dimension also increase. This raises the question of what's the underlying geometry that induces all these three trends. So to understand that, we built a toy model of neural tuning, which is also a Gaussian bump. So this model has two parameters. One is D, the number of ones on the precision matrix rotation, and it signifies the number of tuned dimensions, and the other dimensions are untuned. The another parameter is tuning width, sigma, which specifies how fast it falls off from the peak. So we will see how these two parameters affect the experimental results, like evolution, manifold, or reduced dimension evolution. To give you an intuitive 2D example, we plot 4 versions of tuning maps with 4 different parameter sets. In this case, you have one tuned dimension, which is this, and one untuned dimension, which is flat. And in this case, you have 2 tuned dimensionalities. We see that if you have fewer tuned dimensions, a random subspace can easily intersect this manifold of peaks. And so you are able to search for a peak even in a random subspace, but you're not going to have that for a higher dimension tuning curve. And we also see that if you have a broader tuning, or you have fewer tuned dimensionalities, it's faster to using evolution to optimize it because you have fewer things to optimize. We perform experiments on this parameter grids of tuning width and tuned dimension numbers. And we plot the results as a function on this phase plane. We see that if from early visual cortexes to higher visual cortexes, there's a decrease in the tuning width and the increase in the tuned dimension number, we can recapitulate all our observed results from V1 to ITs, also from the in silico visual hierarchies. Like the search convergence time will increase, the tuning width will decrease, and also the gap of reduced dimension evolution will increase. As a conclusion, we show that evolutions can actually find a local optimums on the high dimension tuning landscape of neurons. And with proper sampling of stimuli, even higher level neurons can show smooth bell-shaped tuning maps around their peaks. And we also think the ramp-shaped tuning or bell-shaped tuning could be just 2 views of the same mountain, and we can discuss that in the final section. And the tuning landscape of neurons generally follows a progression along the visual hierarchy. The higher visual cortical neurons exhibit sharper tuning peaks, and those also have more tuning axes or higher intrinsic dimensionalities. To the end, if you still remember my title, I said the geometry of generative models in brains and machines. So you may think I'm cheating that you have seen the generative models in machines, but it's not in the brain. We just use it to study the brain. So here I would have a short discussion of maybe we can consider the brain itself to be generative. And the link is the so-called energy-based models. So one way to parameterize and distribution is to define the energy function in that space. And then to sample from it, we can optimize energy function and plus some noise, which is Langevin dynamics. We think in our study of the neuron or neuron-guided image sampling, we are doing something very similar. So energy function for energy-based model and tuning function for neurons, and density of energy-based models and density of neurons defines the distribution in the image space. And sampling of the neurons is through the evolution experiment, which is approximates a noisy gradient ascent. And we can interpret neurons as modeling a distribution of images that elicits the highest firing rates. So we think the visual neurons can be considered a generative model like EVM, and evolution is a way to sample from them. So we call this a generative understanding of visual neurons. In the future, I will proceed on 3 different directions. The first is to continue the dissections of inner working of generative models, and specifically like diffusions, transformer, and so on. And we also continue the generative understanding of visual representations and extend our paradigm to population coding, and also study different generative models as priors, and also compare neurons to models using the generative model perspective. And finally, we also want to extend the evolutionary approach to help other scientists to study behavioral neuroscience or perceptions. If you're interested, I can introduce a few sample projects on each of these directions. But for the interest of time, and here's my acknowledgement, and thanks for your attention.
Thank you. So let me read out the questions, and maybe I'll read the first question, and there is a second question from a two-part question from George, which maybe while I'm reading, Elise can add George to allow him to talk. So you can ask it himself. So there was a question from the audience, is whether the fact that is there a relation between your Gaussian theory and the Gaussian part of the diffusion model, the fact that diffusion model itself kind of uses a Gaussian noise and generally is a diffusion from a probability distribution into a Gaussian distribution.
Yeah, that's a very good observations. And I didn't notice that until I make this slide and make this talk, and find the connection is maybe deeper than I think. So like diffusion model is like traveling on the landscapes of, guided by learned approximation of the gradient, which is a score function. And EBM is climbing the gradient of energy function, which is learned by neural networks. And in the neural guided evolutions, you are kind of climbing the landscapes defined by neurons, but using just the evolution algorithm to approximate the stochastic gradient. So in some sense, yeah, like this is kind of a diffusion-like process, which generates guided by the neurons. Yeah.
Thank you. George, do you want to ask your question?
Sure, thanks. Binxu, great talk. I have one minor detail question and then maybe one more general question. The minor detail was for the super stimuli that you're generating for neurons in different parts of the visual hierarchy. After you've evolved a very effective stimulus, have you explored whether there are what the invariances are? So, for example, simply rotating or rescaling those stimuli and seeing if the neurons are robust to those changes, or more so at deeper layers?
That's a super good question. And to be honest, we didn't do the rotation. Mm-hmm. Yeah, we did some sort of translations of the images. And we even do the harder version, which is translated and evolve again and see if we can find the same feature in different positions. And they seem to work. Yeah, but rotation, I don't know if it's robust to it, and we don't have data on that yet. One maybe small comments that for the invariance, like we think our data from the manifold project may shine some light on it, like because on the tuning maps we find you have a peaks, but you can also draw contours around the peaks, and if you visualize the contours, so it's by definition empirically invariance the neurons have to the stimuli. And if you visualize this contour, sometimes you see intriguing invariance like this one, which is kind of like a color circle in the image space. And so in some sense, this neuron is invariant to this color transformation and more interested in the shapes. Yeah, so we think maybe there are ways to use our approach to empirically find what things neurons are really invariant to, instead of like using our priors to test it. But what you said is very interesting. Yeah.
I have another question, but I'll wait and see if other folks want to ask first, I guess.
Sure. So maybe I'll read. There's a short question by Sheng Yang, which is the question is, how do the optimized images depend on the initialization of the evolution?
Very good questions. So we don't have many experimental data on it. Yeah, so in like in experiments, you really start from a setup, and if it works, it's you just keep it. For in silico, we find that so if you use common evolution algorithm, you'd better start from the center of that universe, which because the image manifold is like spherical, so you better start from the center. If you start from like the sphere shell, it doesn't quite work unless you use some special optimizers like spherical evolution algorithm, which I developed previously. And for in vivo neurons, we tried a spherical optimizer, which start from the shell and optimize along the sphere, which also worked. So we think it's so as long as you respect the geometry of the image manifold, it should be fine. Yeah.
Thank you. And maybe George, now you can ask the second part of your question.
Sure, it's real open-ended. So I just was curious to hear how you're thinking about, your work focused on individual units and individual neurons and finding the sort of maximal stimulus for those. But I was curious to hear how you're thinking about a broader population of neurons and whether that taking multiple neurons into account changes how you're thinking about geometry, or whether it's a simple extension of the ideas that you've already developed.
Yeah, that's a very good point. And so currently we think like this energy-based model framework is can like incorporate that easily. So if you have a population code, so you imagine this like vectors, then you can define the energy-based model by multiplying like another vectors on it and define like the inner product or something as the energy and maybe exponentiate it somehow. So experimentally, we have also actually tried that. So you can use your population activations and multiply that by a weight function to design your score. I mean, design your, not the score function score, but like the scores you're interested in and optimize that. So you can sample from that distribution defined by the population code. So as many people doing representation geometry, like you may be interested in see what are the principal axes of this representation manifolds, like maybe PC1 of it is representing something, PC2 is representing something else. And that is a direction we are moving into both experimentally and also in silico. So we have experiments on that.
Great, thank you.
Just a brief comment, and also think that this question is also important for generalizing these tools to broader audience in neuroscience, because people are having larger and larger like recording arrays and more neurons. You definitely don't want to do evolutions one by one. So you may want to do something like based on population structures or correlation structures.
Any other questions? Okay, so if not, let's thank the speaker, and we'll have a short 5-minute break before the next talk. Thank you. Super interesting talk. Thank you very much.
Thank you.